Hello everybody, and welcome to another video with me, Ro, from Scalpel and Stem. In this video, we're going to be doing tissue culture hardening with a twist. I'm going to be experimenting with 6 different substrates to see which one hardens my plants the best. So let's get started. So for this experiment, I'll be using a clear 6-cell tray with a clear catch pot and a clear lid. I've already put my 50-50 peat perlite mix in one of the cells of this clear tray. The other substrates we'll be using are pon, sphagnum moss, small pebble clay or leca, stratum, sometimes called fluval, and small pebble perlite. When using moss, you want to rehydrate it, and since it's tissue culture, I put it in distilled water. So we'll let that soak for a minute. Pon doesn't need rinsed, so we can scoop it directly into the cell. So leca, perlite, and stratum all need rinse to remove the dust particles. Now that I've let the moss sit and rehydrate, I want to make sure to squeeze out the excess water before putting it in the cell. And here we are. We have our cells loaded with a 50-50 peat perlite mix, the small clay pebbles called leca, stratum, sometimes called fluval, sphagnum moss, small pebble perlite, and pon. I've rooted these Periso Verde in a media that contains activated charcoal, which is why my rooting media is almost always black. So first, I'll remove my cultures from the jar. And I'll place them in this jar of dish soap and water. I'll mix it around a little bit and let it sit for 10 to 15 minutes. So I remove my cultures from the soapy water and take off any dead tissue that I can. Additionally, there may be some media left, and you can gently pull that off as well. And then I place my culture into the solution of 1% bleach and tween 20. I've let these sit in the bleach solution for about 15 to 20 minutes. And I'm removing them from the bleach solution and putting them into a jar of deionized water. This will help rinse off any bleach and make sure that you don't get chlorine uptake in these fresh little roots. After letting my culture soak for about 15 minutes in water, I then transfer them to Fizan. This helps to combat any fungus or bacteria that might arise. Now that I've soaked my plants in Fizan, I'll plant them in the tray. Oh, big boy. Here is our tray of Periso, all potted up, with the dome on and closed. Thanks for watching my video. Comment below on which one you think is going to work best, and I'll update in a few weeks. Bye everybody.